Okay, so in addition to infrared spectroscopy, we also have a technique called mass spectrometry, or simply MS. Note this is spectrometry, it is not spectroscopy. It's a confusing distinction, but one method, spectroscopy, relies on photons and light, and mass spectrometry typically does not involve photons or light detection. So this is another method for elucidating a molecule structure. Probably the most important information you get from mass spec is you'll generally be able to learn your molecule's molecular weight. And that helps you understand how big is my molecule going to be. I think my molecule has 20 carbons. Well, if you find out that it has a molecular weight of 100, it definitely doesn't have 20 carbons. So it gives you a sense of the size of the problem. In some ways, mass spec will also reveal what some of the parts are of your molecule, and that can also be helpful as well. We won't go through really the physics of mass spectrometry, but what happens is a molecule enters the mass spectrometer and it is ionized. It loses an electron, and typically this electron will be lost from a lone pair of electrons. So you go from having a pair of electrons to having a single electron. When that electron is lost, you form a cation, and it also has, since it has an unpaired electron, it's called a radical. So we'll form a radical cation. That's supposed to be an A, if you can't tell, radical cation. And this radical cation may pass through the mass spectrometer intact, in which you will detect the entirety of the weight of that molecule, or it might break into pieces. It'll undergo fragmentation. And when you observe these pieces, that's where you start to get a sense of what are the parts of your molecule. So let's go to the next slide and see this process again. So here's our molecule. This molecule has a lone pair of electrons. You don't have to lose the electron out of a lone pair, but for our molecules, I'm trying to think of an exception, and it's not. We're going to lose them from lone pairs. We ionize. When we ionize, we will lose an electron, subtract out an electron. So necessarily, the parent ion will have a positive charge. It loses a negative charge, it must have a positive charge. And it has both an unpaired electron, so it's a radical, and it has a positive charge, so it's also a cation. We'll call this the radical cation. Sometimes we'll detect the parent ion. When we detect the parent ion, it has an M to Z, that stands for mass to charge ratio. Typically, Z is going to equal 1, so the M to Z ratio is simply going to be the mass of the cation. In this case, the molecular weight of this parent ion is the mass of this parent ion is the same as our molecular weight. Now, these radical cations sometimes are unstable and they'll fall apart further. They'll fragment, and we'll get pieces. When you form your fragments, one fragment is typically a radical, and the other one is a cation. When we do mass spec, what am I watching fragment for? Words are difficult sometimes. Okay, so we get a cation and we get a radical. When in our detector, we detect the positively charged things. So we detect the parent ion that has a positive charge, and we can determine our molecular weight. In terms of our fragments, we will only detect the cation. We will not detect the fragments. So this gives you a sense of what happens in a mass spectrometer. We lose our electron, we undergo ionization, and we get cations, radical cations, and then sometimes these fragment, and we'll detect all the positively charged species. So let's get some general ideas. When we ionize the molecule, it almost always occurs for us, in fact it will always occur at a lone pair. And the lone pair could be on an oxygen, a nitrogen, or halogens. If your molecule contains multiple oxygens or nitrogens, fragmentation can occur at any one of those spots. It won't occur at multiple spots, but any one of those spots for a given molecule. Once we form our parent ion, our radical cation, it can fragment, break into pieces. And I've put the prime there to show that this fragment is going to be different from the other fragment. The pieces don't have to be equal halves. You can have small pieces and large pieces break off. In particular, one thing that's pretty interesting are when your molecule contains a chlorine or a bromine. So I'm talking about molecular weight. When we think about molecular weight, that's the average molecular weight for our species. But chlorine and bromine are present in significant quantities of different isotopes. Chlorine is chlorine 35 and chlorine 37. And it's about a 3-to-1 ratio, a 75-25 ratio of that. So when you detect a molecule that contains chlorine, some of those molecules are going to be chlorine 35, and some of them are going to be chlorine 37. And so in a mass spec, these show up as different species because they have different weights. One has 35 as a contribution from chlorine. The other is 37. There's not an average. We're detecting individual molecules. And those individual molecules have individual masses. And so actually we will give rise to different patterns in our mass spectrum. And so I'm just going to plant the seed and say, when we have halogens in our molecules, specifically chlorine and bromine, we're going to see different peak patterns. And those are going to be very characteristics, and they will tell us immediately whether we have a chlorine or bromine in the molecule. So it's not just about molecular weight. It can also be about types of atoms, particularly chlorine and bromine, and sometimes we get fragmentation information from this as well. And we'll see this in the examples.